Hello, my name is Asher Boorstein, and my mentor for this project was Dr. Balui TV Durohe, who is a nutrition scientist. I attend Middleton High School. I live in Middleton, Wisconsin. I'm zooming in from Middleton, Wisconsin, and I'm in class of 2025. And today I'll be presenting on the applications of RNAi and CRISPR gene editing technologies to decrease immunodominant allergens in foods. So to start off, what is food allergy? Defined by Johns Hopkins Medicine, food allergy is abnormal responses to the body's immune system to certain foods. And these abnormal responses, also called allergic reactions, can happen very soon after eating the food, and it can happen in very small amounts of the allergen, which makes it very life-threatening, even in the smallest amounts. And the symptoms that arise from allergic reactions include hives, vomiting, and wheezing, and anaphylaxis. Anaphylaxis is the most life-threatening because it can tighten your airways, and um it impedes everyday life. So I know this from experience because I have a tree nut allergy, and you have to be often extremely cautious when deciding what you can or cannot eat outside of your living area, because one wrong thing you eat, which is contaminated with your allergen, could lead to anaphylaxis. And to not get food allergy confused with food intolerance is less severe. So the prevalence is very high regarding food allergy with children. It's about 6%, and that's about a little over 4 million children within the US who have food allergy, and 6.2% for adults. And this follows heavy economic costs that have factors of outpatient visits, medical drug prescriptions, and emergency visits. And this table on the right is from a cross-sectional survey study conducted with a representative sample of approximately 1,600 US caregivers of someone with food allergy. And the researchers observed the cost of $5.5 billion out of pocket for food allergy per year in the US, or $931 per child. And the total US spending on food allergy was found to be $25 billion per year. So there are quite a few treatment and management methods, not many actually, but so the first one is epinephrine, also EpiPen. Many of you may know this. For someone who has a food allergy, often carries these around everywhere they go. So epinephrine is a naturally producing hormone within your body for your fight or flight response. So when it's extreme times of stress and fright, epinephrine will be released from your adrenal glands right above the kidney. And epinephrine is also used as a management tool for anaphylaxis, which is one of the symptoms of um, allergic reaction to a specific food. And what epinephrine will do is dilate the airways and make your heart pump faster, which increases blood flow. And you will often need multiple doses of epinephrine because there's biphasic anaphylaxis, which means anaphylaxis can rebound. So this anaphylaxis also usually follows with an emergency room visit because you'll need multiple doses of epinephrine to counter the rebound. The next treatment, this is oral immunotherapy, which helps desensitize patients to a specific food allergen by giving the allergic individuals small increasing doses of the allergen for often days, every day for often weeks. And this will help build the body's resistance to this particular allergen of choice. And it was seen in a peanut oral immunotherapy study of this effectiveness that patients in this study for peanuts were randomly assigned to either a placebo or the drug AR101, which is the oral biological drug that contains the peanut allergens. And as a result, the patients who received the active treatment were able to consume more milligrams of peanut protein, as well as having a less severity of allergy symptoms when eating the allergen. And this process can be very tedious. And often when you stop doing this process, the desensitization effects will often diminish. So So to begin with the biology of food allergy. So first, the body identifies the substance allergen as foreign. In this case, is allergen. The antigen presenting cells carry the antigen or the allergen in this case to the T cells, and also called helper T cells, will secrete cytokines, which are chemical messengers, which the chemical messengers will then communicate with the activation of the B cells, which will differentiate into plasma cells, and the final result as an IgE antibody production. And for sensitized allergic individuals, IgE will bind to these mast cells. So when an antigen or allergen is entered through the body, they'll bind to these IgE on the mast cell and release histamines, which are the inducer of the symptoms of allergy. And these are preloaded in people with allergy, so the effect of histamine release will be like instant once the allergy is consumed or in contact with. And IgE production is very important for allergy research because, especially for my presentation, because when researchers want to determine the allergenicity of a specific food or allergen or protein, they will often use IgE binding to test the allergenicity of certain proteins, such. So, what is RNAi interference? RNA interference, also called RNAi. So this is a phenomenon found in C. elegans, which is a multicellular nematode, where small pieces of RNA can shut down protein translation and bind, which by binding to the messenger RNA, mRNA, that codes for the proteins. So essentially, there will be a complementary strand of RNA that will bind to the messenger RNA. So before it gets to the ribosome, it will degrade the messenger RNA. So it will shut down the process of making the allergen, in this case, allergenic protein. So this has been used in agriculture, drugs, plant drought tolerance, and food allergy, and much more. And when unsure about a specific function of a protein, scientists will utilize this phenomenon to observe the after effects of shutting down a specific protein. And this is called knockdown because the genes could still be present for the making of the protein. It will just halt the final production of the protein. Now CRISPR. First discovered by accident by Japanese scientists in the bacteria E. coli. It was discovered as a bacterial defense mechanism in bacteria to defend against bacteriophages. So the bacteria will cut segments of the bacteriophage's DNA that it inserts, put it into its own. So now it has a complementary strand. It can now match up with the bacteriophage's DNA and cut pieces of the DNA as the defense mechanism for bacteria. And within the biotechnology setting, CRISPR researchers utilize the Cas9 nuclease protein, which are essentially nuclear scissors that is guided by single guide RNA and a custom designed sequence that is complementary to the target gene. And this is often used to edit immunodominant allergen genes of food or plants. And this is a form of knockdown, meaning it completely deletes the target gene, degrades it, rather than knock out, I mean. So to completely degrades, knock out, and then knock down, as for RNAi, would just halt the translation of the protein, but not the gene itself. So the laboratory procedure of using these gene editing techniques are fairly similar. They both use the same methods, essentially, and they utilize plasmids, which are located in bacteria, and they're circular pieces of DNA. And researchers can implement like complementary strands of the DNA or mRNA they're targeting into the plasmid. And since they're in a bacteria, they can be put in a bacteria that most of these studies use as atumefacines. And what they will do with the bacteria that has the plasmid with the gene of selection customized, they will infect food cultivars with the bacteria. So the plasmids in the food cultivars can be implemented as complementary strands. And scientists test whether it was successfully implemented by using an antibiotic resistance gene. By observing this gene, they can use um, antibiotic. So when they test antibiotic and it just completely kills all of it, it did not work because there's an antibiotic resistance gene. So it would survive if it was infected successfully. So the results of RNAi, with the RNAi studies I reviewed, so all these studies, soy, wheat, apple, peanut, all the immunodominant allergens of these food were silenced to various degrees. But and all of the phenotype typically observed traits were that there were no major differences in the morphology and reproduction factors between the transgenic line, which is the one that had RNAi mutated on, or RNAi used on, and the wild type, which is the original plant crop. So there were no differences, which is a good sign. And IgE binding was observed for wheat and peanut, which, as I mentioned before, is the IgE binds to specific allergenic substances. So IgE binding was less in the RNAi silence lines than the original ones, which indicated a success in silencing the allergenic proteins. That was observed in wheat and peanut. And skin prick test, which is in vivo and uses allergen on a claw or really sharp point and rubs it into the skin of a patient that is documented with, in this case, apple allergy, and they detect the rash size. So if the size is big enough, they can detect that the wild type had more of a size than the transgenic because of the allergen protein was removed. So as you can see in here, this was an immunosorbent assay that detected a major decrease in the ARAH2 protein, which is the protein for the peanut. So there's a significant decrease, as you can see. And using 5 patients, Sarah, researchers found a significant decrease in IgE binding for each patient. So compared to the wild type, which is the colored shaded in right here, there should obviously be a higher IgE binding for that because it's the original compared to the transgenic lines, which were edited or silenced. And so now CRISPR. So again, for the CRISPR studies, all of the immunodominant allergens were successfully edited, and they not all of them observed the effects such as IgE binding, skin protects, and phenotype, except for brown mustard, which they noticed um variability in the seed weight and reduction in the seed production. And yeah, as you can see, they not all of these used skin prick or IgE binding. And this is the brown mustard study. As you can see in section E, this is the entire process for how they observe the phenotypes. And for section E, you can see a significant decrease in seed production and growth for the transgenic line. And these are bands that represent the removal of the allergenic gene of brown mustard from the cultivar. As you can see from these bands, it was successfully removed. So now we will get to future directions/limitations. So clinical trials are needed to measure the safety and efficacy for actually benefiting people within society who have food allergies. So yes, we have the results of these papers that show that we can significantly reduce IgE binding from editing or silencing allergen genes, which as a result will decrease allergenicity for food allergic patients. But only 3 studies reviewed looking at IgE binding, and one of them used in vivo, which was the skin prick test for the apple study. So that is something to look into. And not all food varieties were experimented. So there are hundreds of varieties for multiple food cultivars examined, such as soybean, and some of them could have more or less IgE binding than others. And ecosystem and societal acceptance is something to look into when silencing and editing crops that we would potentially use in the ecosystem possibly, or it would possibly spread within the ecosystems, and cultural acceptance would also be a factor to consider. So thank you for listening to my presentation today. Hope you enjoyed.